How is everybody? What class is this? Is this AnP 2? The one thing I haven't really looked at lately is what room I'm in. So. And I thought this might be like 1108 or something. Talking about turtles. Alright, so I scared you. So everybody good? Yeah. Kind of our first class-ish, but not. Hurricane storm of the century. Wow. It's supposed to be windy tomorrow, so who knows? We have classes then too. We might get used to this. Alright, so what? We're Tuesday, Thursday, in person, 15 weeks. Everybody cool with that, right? Alright, biology 2252. 22 lecture. So sorry, kids fighting for the internet and trying to not sit on a Godzilla toy or something. I tried to walk through in the Zoom with horrible internet access, as you can see, probably jumping video, but if you can stomach that one, just make sure you look through that one, because that's really where I focus on polio in that video link that I give you access to. I walk you through the syllabus, I walk you through the expectations, I talk about the way the course is going to be broken down, and the chapters and the book and all that that we use. Everybody's cool with that? I can pretty much jump in. So in Folio, all I've done is come over to the content section, and I've, since we've spoken by Zoom, I've uploaded really all the content from the first chapter. Now, in a perfect world, we would start off with chapter 16, which is kind of the continuation from AnP 1. So you can take it with the biology department, you can take it with kinesiology here on this campus, or you can transfer in with AnP 1. But typically AnP 1 runs through the muscular system, skeletal anatomy, tissues, organs, and then get into nervous system, special senses, etc. Chapter 16 was the endocrine system, and that technically is the first chapter of our textbook that we would probably normally be picking up on. But a lot of you are in lab this same semester. And the lab, especially biology version of it, 2252, the labs, the sequence gets all screwed up if I start with endocrine system. So I'm starting with the blood. We'll come back to the endocrine system, chapter 16, at the end of the semester. Okay, so I mentioned that little video thing, but I just want to make that clear in case anybody's kind of wondering why we jump in like this. So without further delay, I'm out. Hi, how are you guys? Office hours, you know, are posted. You can find me down the hall. I can't help but say some of the introductory stuff. I'm in, I got 1308, I think, down the hall that way somewhere. So you guys can find me at any time if you need to. So let's think about the blood. And listen, sometimes in these little column slides, column whatever, this one was based on the Word document, obviously, with some images that I didn't dig in here. But sometimes I'll just have headers for you to consider. And so, I even mentioned, you might want to write a few examples down. So if I mention the blood, and we're going to start to look at fluid that travels through our arteries, veins, capillaries, etc. If I mention transportation, I'm asking you now, tell me something that floats through your blood, through my blood at any given moment. And I heard something. Go ahead, hit me. Oh, God, we're scared of each other, aren't we?
Minerals.
Minerals, yeah, we've got all kinds of vitamins, we've got all kinds of key elements that flow through the blood, right? What else goes through us? Oxygen, right? And if you say oxygen, think about the flip side, carbon dioxide, right? So we have some of these respiratory gases that are floating through. Now, some of these are going to be bound specifically to the erythrocytes and hemoglobin. We'll talk about that more specifically as we go, but excellent for both of those. What else floats through my blood? What's that? Exactly. So urea, uric acid, even a little bit of ammonia got to help us. Some of these noxious compounds, a lot of excess metabolites, pharmaceutical drugs get broken down by the liver, right? They get routed through the bloodstream for a period of time. Go ahead, I'm sorry.
Immune cells?
Yeah. So those are the white blood cells, right? Which is a big focus of this chapter. And if you're in lab right now, listen, there's dueling labs. One starts 7 weeks in for Mini-Semester 2. Some of you are with me or Dr. Mans or Trum, and you guys might have AnP 2 right now. But that's a big focus of one of your first lab exercises, identifying those different white blood cells, which can be tricky. We'll introduce those here shortly, but yeah, exactly, immune system function, right? Keep us healthy. Also, things like our electrolytes, like sodium, potassium, calcium, magnesium, and you have to write all these down and scramble. You really don't, but I'm saying, if you want to jot a few thoughts down to yourself, you can, but you just kind of keep tabs of what we're describing. But lots of things, hormones, you with me? Like over here I say the word regulation, and if you think about regulation, think about your growth, think about bone deposition, think about the increase in muscle mass if you're in weightlifting this semester, as part of your PE requirement, right, college or just something you do on the side. You're gonna need hormones to kind of regulate growth and development, reproduction, right? Calcium homeostasis, right? Blood sugar levels, insulin, there's all kinds of things that are involved in, so we're talking transportation, I just kind of zoom in here to regulation. So hormones that flow through the blood are transported, that they help regulate our body, maintain homeostasis, right? And coordinate development, growth, reproduction, all these kinds of things. Regulation also, in these few days where it gets, dipping down towards, the freezing mark at night and through the early morning hours. You go out, you're changing the tire, you're stuck outside, maybe you work, in and out of, some setting, and you're in the cold for a period of time. Sometimes when you come in, you will feel your ears just start to radiate and become incredibly, hot. Maybe may even turn kind of like a bright red. That's thermoregulation. So our blood can actually be shunted away from tissues that are kind of superficial, like your nose. Now, tips of my fingers are very important to me, but they get pretty cold too. But just think about like your fleshy ears. You with me? If you radiate blood there, I say radiate, let me see if I can explain it better. If you have blood flowing, right, through the tissues of your outer ear, your pinna, and the skin, it's going to radiate heat away from the body when it's cold on these mornings like we've had the last few weeks. And I'm saying to you, what happens is that little capillaries and blood vessels just squeeze shut, and so you don't allow that blood to travel so superficially because you don't need to lose it. You with me? You don't need to just radiate it away from the body. When you come back in, those vessels can relax and allow blood to finally travel through these tissues of the ear, and that's where it gets really, really hot to the touch. Well, that's regulation, you with me? So even regulation of your body temperature can be there as well. Again, we're just trying to spitball a few things here. We're not trying to be comprehensive. Protection, let's just go back to immune system. So your white blood cells that we'll see here shortly, the different classes that we have, they have different functions, different actions, and they're gonna keep you healthy. So we'll keep it kind of vague. All right, so what is blood itself? And I'll avoid quizzing you this first day too much, but in time I hope you guys will, of course, interact. You already are, I don't mean it dismissively, I just won't overdo it today. But if you remember, blood's a weird tissue. It's a connective tissue, but it's a fluid, obviously. And a connective tissue, if I think connective tissue, I think about like cartilage, I think about bone, I think about some of the tendons and ligaments in the body. That does not seem like blood. But the common characteristic they share is living cells embedded in, an extracellular matrix, a non-living matrix, a ground substance, you use different terms for it. But in blood, the fluid, the water-based plasma, is an acellular, it's a watery mix, it's not living, you with me? But the living cells, white blood cells, red blood cells, and platelets float within it. You got me? So that's kind of the basic definition of connective tissue. Living cells embedded in kind of a non-living matrix. Here, the matrix is plasma, a water-based solution, So 8% of your body mass, do you need to know that it's 8? Will I ever ask you, is it 9, 10%, 11%? No, I will not, you with me? But it does represent a sizable overall mass when we step on the scale. Between 4 and 6 liters, depending on sex. Again, you don't need to tell me the exact number of liters of blood, but just to give you kind of a ballpark, right? 5, 6 liters typically in males, maybe a little less in females on average. Slightly alkaline pH, 7.35 to 7.45. That is not a number you need to memorize for the first test, but in time, as we go this semester, we're going to talk about our pH of our body fluids, including the blood. And this is going to be something we're going to come back to. We regulate very carefully the pH of our fluids. So again, little factoids that you see here that seem very specific. I'm not trying to trip you up on a future test. We're just setting the stage, okay? And I'll make it clear the specifics you do need to know as we go. And you guys can always ask if there's any confusion. So if you take a sample of blood and you centrifuge it at high revolutions per minute, right? I'll spin down 5-10 minutes or so. You will definitely see a separation in the sample. You're gonna see a heavy layer, which is gonna represent about 45% or so of the whole. This will be your packed red blood cells, your erythrocytes that settle to the bottom, being fairly heavy. Most of what you find, about 55%, would be kind of a lighter straw colored, slightly amber colored fluid. That would be the water-based plasma. And there's lots of things that float in plasma. And we were mentioning things that float in the blood, transported and stuff like that. So we'll come back to that, what is in the plasma, what makes up plasma in just a second. But notice you also get kind of a little haze region right between the packed red blood cells at the bottom and the straw-colored plasma at the top, and that's sometimes described as the Buffy coat. And it's less than 1% of the overall whole, so most of what you see is plasma, water, and red blood cells. But about 1%, a little less, is called the Buffy coat. It looks a little hazy if you look real careful. You got a big mixture. And that's where all your leukocytes or white blood cells and your platelets are found. Okay? So, just give you some bearings. All right, so let's move on down here. Everybody see it okay? And there is no beauty to my slides. Most people will do these in PowerPoints. They're just giant, and I sometimes think a little more font, a little more description on here. Sometimes I just do the word stuff. All I can say is you've got this. You can download it, you can print it for yourself. So I apologize if you don't have your device with you, you're relying on me. You might have to squint a little bit. I can always try and zoom in, and I will at times. But I'm hoping you guys will either print them out before you come in, old school, or just pull them up as you're working today, so you can see them a little better. If you're squinting, I don't have the best vision. I feel for you. But let's not let that get in our way. All right, so what is plasma? It's about 55% of the whole sample, right? It's a water-based solution, which is well over 90% just H2O, water, okay? There's all kinds of dissolved solvents, and I say provide a few lecture examples for the following. So electrolytes, just hit a few here. Sodium, potassium, magnesium, calcium, chloride, you with me? These are the things that power action potentials. We think about the nervous system, communication, muscle contraction, all these kinds of things, like calcium, nerve transmission across the synapse. Okay, involves calcium and the release of little neurotransmitters. So sodium, potassium, chloride, calcium. Don't worry, but those are really important things that float in the blood that we're going to need for our nervous system, for muscle contraction, for our cells to function. Nutrients, all the things we'll talk about in a few weeks when we get to digestion. So all the carbohydrates that we consume, the proteins, right? The fats and the lipids, even the DNA and RNA in the food that we eat, right? Whether they're plant tissues, potato chips, French fries, you with me? Or a chunk of steak or a hamburger, something like that. You're bringing in a lot of DNA, a lot of RNA from these living organisms that we consume, okay? Look, all those get broken down into their little chemical ingredients. So proteins get broken down into amino acids, and carbohydrates get broken down into little simple sugars like glucose and sucrose and things. We're gonna absorb those and suck those into the blood, you with me? So we have a lot of nutrients that are going to flow through our bloodstream to be delivered to brain cells and muscle cells and all the different tissues and organs of the body, okay? So nutrients, just put glucose, amino acids, lipids, don't worry too much. This is not for test purposes, like I'm going to put a term like this and have you give me 5 examples. we're just chatting to get started. Gas is real simple, oxygen, carbon dioxide, let's just focus on those are the biggies. Hormones, do you need to write down a lot of these hormones? No, but think about it. Chemical messengers, that's what a hormone is, released by endocrine tissues, glands, and cells through the body. Hormones are things like testosterone and estrogen, sex hormones, right? Growth hormone, thyroid hormones, you with me? So these are the hormones that orchestrate our growth, our day-to-day survival, big events like reproduction, times of puberty, menopause, what have you. You get changes in the levels of hormones that are being secreted. So nothing you have to memorize forwards and backwards. Waste products, we had talked a little bit about these. If you want to, just hit the biggies like urea, for example, ammonia, uric acid, things of this nature. And there's so many others, but that's a quick round. Plasma proteins, this is where it gets a little, not confusing, but maybe this is less obvious to us. You have proteins that float in your blood at all times. They float in your plasma. They are dissolved, floating in water-based plasma, and they travel through your arteries and veins, getting pumped around the body by the heart. And they're just proteins, but because they're in the blood, because they float in plasma, we call them plasma proteins. And this chapter, we'll talk about a bunch. So we'll introduce a bunch of these by name. And through the semester, every system, every chapter, we'll be talking about proteins that float in your blood that are going to trigger clotting if you have a rip in a blood vessel. I won't mention all these right now, but before we're done with this chapter, we'll talk about 7, 8, 9, 12, you know what I'm saying? But for now, just as an example, albumin is a little protein that floats in your bloodstream. It's the most common small little protein that floats in your blood, albumin. Albumin actually is like a little Uber or a taxi. It's like a little shuttle protein. And it can carry little materials along with it through the blood to help deliver them to different organs and cells and tissues. So no test question on albumin and what it does, but I'm just saying we'll look at a lot of these little plasma proteins as we go. All right, listen, I'm gonna say again and again, white blood cells, red blood cells, and platelets. But if you really, really wanted to be specific, you might use the term formed elements. And I'll say formed elements, and then I'll probably just call them cells for the most part. So white blood cells, red blood cells, and platelets, or thrombocytes for platelets, leukocytes for white blood cells, and erythrocytes for your red blood cells. They're all cells. The thing is, some of the cells don't seem to be complete. So if you go back and look at chapter 3-ish, maybe, from your AnP textbook, we're using the same text. I feel like I've robbed you of something that I expect you to go watch a video, but that's what I'm gonna do, you with me? So I've explained their textbook and layout of it, these kinds of things online for you. But it would be back in a previous AnP course, whether you transferred in or took it with Craven or somebody down the hall last semester perhaps, or earlier. You get into cell theory. All cells have a nucleus, all cells have mitochondria and cell organelles and Golgi bodies and blah, blah, blah. What I'm saying to you is that some of the cells here aren't complete. Like erythrocytes, when they float through your bloodstream, they don't have a nucleus. So they lack a nucleus, you with me? Little thrombocytes are just little cell fragments, little bits of a membrane with cytoplasm inside and clotting proteins. So there's no nucleus, no organelles, you with me? So I'm saying to you, a lot of people stress that these should be called formed elements rather than cells, because some of them are kind of incomplete when they float through your bloodstream. So formed elements is basically the same thing as calling them red blood cells, you with me? And I'm gonna call them red blood cells and platelets and blah, blah, blah. But some of them are not complete. All right, so that was up here, I guess. All right, so erythrocytes, these are the biggies to start off. These are your red blood cells. Red blood cells are pretty small. Now, you see the little micron or micrometer symbol here? So, you know, fractions of millimeters in length. Not a negative, but a round little about symbol there. Not anything that I need you to memorize hard numbers here, but just as a reference,
Okay, about 7.5 microns is the diameter of a very, very tiny cell, which is so numerous in the blood, and that would be a red blood cell. So we'll make sense of the 7.5 microns. For now, I'll just simply say it's about the same diameter as some of your smallest little blood vessels called capillaries. So these things can, in some cases, some capillaries are bigger, some capillaries are more narrow. And some of the tiniest little capillaries in the body, right, 7.5 microns is just enough to kind of squeeze through single file. So these are almost too big for some of our tiniest little blood vessels, but they are tiny, and we'll kind of make this reference as we go on. So 7.5 microns, they're very specialized cells. We can call them anucleate, like to lack a nucleus. They don't have a nucleus when they're floating through your blood mature. And we'll talk about how they're produced. They have a nucleus for a period of time developmentally, but they shed the nucleus just towards the end, and then they're going to float through the bloodstream for a period of 120 or so days. If you don't have a nucleus, you're doomed. You can't replicate, you can't divide. So these have to be produced constantly from stem cells in our bone marrow, which we'll talk about as we go, okay? So mature, they have no nucleus. They're also biconcave, they're kind of pinched, right? So they're kind of pinched from the top and the bottom, kind of an inner tube kind of pinch, if you will, but still not hollow, right? But kind of pinched inward. So biconcave shape. And the biconcave shape is actually because of a little protein, kind of a cytoskeletal protein called spectrin. That's not a term I'm going to ask you to name. I don't know that spectrin is the most important protein. It is a protein, but it's kind of like a little suspension cable on a bridge. You with me? And it's going to crisscross the interior of the cell, kind of holding it tight so it has that biconcave shape. Why make a big deal out of this? Well, first of all, it's just how it looks, so that's a descriptive thing. But that pinched little effect, that biconcave shape and no nucleus, allows them to have the best surface area to volume ratio, which is a fancy way of saying they can load the hell up on oxygen as they pass through little capillaries around their lungs. When we take a deep breath in, an inhalation, these little guys are moving right next to the little microscopic air sacs, alveoli, of our lungs, and oxygen can just flush in, and it can bind to little proteins called hemoglobin inside the red blood cell. So the biconcave shape is part of their brilliance at carrying oxygen and later releasing it to the tissues of the body, which is something we'll come back to later on. So the biggest protein here that you do need to know will be the hemoglobin protein. So many millions of hemoglobin proteins packed into every red blood cell. Hemoglobin is important enough because it's the one that's going to participate in oxygen, right? Loading and unloading, if you will. So oxygen, O2 is going to bind to hemoglobin, which is kind of part protein, part of a little heme group, and we'll come back to that. Remember how I said that if you centrifuge blood, that a large component of it is this ruby red that settles to the bottom? I said those are the packed red blood cells. There's a term for this, that packed red blood cell. I said is around maybe 45% or so, typically on average. You can call it the hematocrit. So if you hear the word hematocrit, go back to the little test tube. I won't scroll up, but if you go back to that test tube with the separated blood, that level of 45-ish% of the packed red blood cells, that would be your hematocrit. And the hematocrit is really a kind of a percentage measure based on the whole sample. So a hematocrit of 45% or 42% is really saying 45% of your whole blood was red blood cells. You with me? So hematocrit, if you hear that, it's just kind of a representation of the percentage of your red blood cells in a sample of volume of whole blood. Okay, the hematocrit. I mentioned here it varies a little bit, so between 47 and 42%. 45 is typically the average that you'll see in most illustrations, most figures, right? Just kind of looking at everyone. We took an average of our hematocrit, it'd probably be around 45%. Might be a little lower in females, might be a little higher in males, but that's not anything I'm too worried about for lecture purposes. Hemoglobin is going to bind oxygen. Now, hemoglobin HGB is how I always abbreviated it. That's how I was always taught. Some book authors, I think yours do now, just go HB. It does not matter, but yeah, HGB for hemoglobin, HB for hemoglobin. So just an abbreviation you may see in my slides as we go a little further. So what is hemoglobin? It's a globin protein. So part of it is a protein, the largest component, which has 2 alpha and 2 beta polypeptide chains. Let's take this in for a second. So proteins are made of little amino acids, right, that are all kind of bound together, right? Peptide bonds all stacked together like pearls on a string. And the amino acids, pearls on a string, are then kind of folded up. And if you have lots of amino acids, you may use the term peptide. It's like a fragment of a protein. That's what you're building. So lots of amino acids, or some amino acids, you may say it's peptide. If you get a lot of amino acids, you may just loosely use the term polypeptide. So we basically have 4 polypeptide chains, but 2 are identical. We call those 2 alpha, based on their orientation and arrangement of amino acids. And then we have 2 beta polypeptide chains. Again, what are these? Just a bunch of amino acids all glued together. Okay? So put those together, and we get a globular looking protein called the globin. You can see the 2 alpha in purple and the 2 beta in blue. Those would be those polypeptide chains. So the blue and the purple goo that you look at there, that globular thing, that would be the globin. You with me? But notice that in each of these polypeptide chains, we have one of these heme pigments, which contains a little Fe with a little 2 plus there. What is that atom there, Fe?
Iron.
Yeah, iron. So we have a little iron atom at the center of a little heme group. So that will be centered on each of the 4 polypeptide chains. And it just so happens that oxygen molecularly likes to bind really tightly to this little iron atom. So oxygen at times will seek out and bind tightly to the heme group, and you got 4 of them in a polypeptide chain. So if oxygen is seated at all of those for one hemoglobin, you may say oxyhemoglobin. But we find it's a reversible process. So at times, the red blood cell is carrying all the oxygen it can. So hemoglobin, right, is carrying oxygen, it's bound to. And all the millions of hemoglobins, proteins in this red blood cell would all be packed with oxygen. We might say loosely it's oxyhemoglobin. But of course, usually we're not looking at one red blood cell with millions of hemoglobin proteins. We're looking at a liter of blood, or a pint of blood, or a test tube of blood. So instead of saying oxyhemoglobin, we're more often going to call it oxygenated blood or deoxygenated blood. But it's really, are you carrying a lot of oxygen on your heme group, or are you not carrying much? It's a reversible reaction. So as we go by the lungs, we'll pick up oxygen and release CO2 into the air sacs of the lungs that be exhaled in a moment. Freshly oxygenated blood now goes back to the heart, gets pumped systemically, and when it gets to the capillaries of the big toe or the liver, we're going to watch a reversal. Oxygen will be released from hemoglobin and go out to all the cells in need, while CO2 enters the red blood cell, and then we repeat the process. More on all that later, but it is reversible. So I think I've covered this to the gist that we need to, I think, right now. I will tell you that that it does matter, and I don't think I have a blurb about it right here. Yeah, I don't. I'll just tell you, it's not written down, but real quickly, if you take a sample of blood from a typical vein of the body, right, a systemic vein, and you take a sample of vial of blood, right, a clear test tube, each one, from an artery of the body, the arterial blood typically is going to have much more of a ruby red coloration to it. That is loaded with oxygen. So when we say heme pigment, it is a pigment because a sample of blood where you have a lot of oxyhemoglobin, meaning oxygenated blood, coming from like a typical artery, that is going to be ruby red. When you look at a sample taken from a vein that has less oxygen, it's going to be a little more of the maroon kind of red, kind of a darker off-red color, if you will. So indeed, heme, I say, is a pigment. Pigment, I mean that when light strikes it in a test tube, let's say, right, and bounces back on our retina, we perceive different ranges of color. Okay, so oxygenated blood is more ruby red. That's not a test question per se, but it is something we'll kind of highlight as we go a little this semester. It just wasn't written down here, and I'm sorry I don't have a little, a test tube looking back at you with oxygenated and deoxygenated blood. Minor point, if you will, but interesting, and something that we'll touch on when we get to respiration. We talk about this cycle of exchanging oxygen, CO2, the tissues and lungs. Real quickly, in terms of red blood cell life cycle, we'll come back to this a little bit with the digestive system, but here's the highlights just so you know. These red blood cells, erythrocytes, if you have no nucleus, you're produced in bone marrow. Once you enter the blood, like I said, you're gonna live for a period of time, a few months, about 120 days, and then you're gonna start to peter out. You'll start to become a little abnormal, you start to break down a little bit, you with me? And so after about 120 days or so, red blood cells aren't doing their job like they used to. And they're constantly being produced in bone marrow, like we'll talk about later this chapter, so we're gonna pull them from the circulatory system. This happens in the liver, and it happens in organs like the spleen, and we'll look at this in its glory later, on in the semester. Okay, but cells, we'll pull them, phagocytic cells, macrophages, other white blood cells, will rip these apart, gobble them up, the old, aging red blood cells. And then we're gonna do our best to rip apart the globin, protein, the polypeptide chains, and hold on to those amino acids. Don't wanna lose those. We can rebuild hemoglobin with them down the road. We're gonna pull the iron atom, because iron comes from, supernova explosion. We don't make it, it's from the stars. Oh my God, this just got deep. Are we all smoking the same thing? We're all made of stardust. Oh, please, I'm gonna sit down, I'm lightheaded. Yeah, so we need that iron, you with me? So we're going to hook that iron to a little shuttle protein, a plasma protein called transferrin. And transferrin is gonna take that little iron atom from the liver, from the spleen, where we've ripped it apart from old red blood cells. It's gonna take it back down to the bone marrow so we can make more red blood cells, more hemoglobin, you with me? So transferrin will help us route iron. Iron is kind of toxic to the body, so you don't want those little atoms just floating outside in the plasma, okay? So there you go. So we use it again. We try and recycle is a big thing. But we do get some of that discarded kind of circular heme group, if you will. You saw those bonds and carbons and hydrogen and stuff. Don't worry about the chemical signature. But that heme group, now that you've stolen iron from it, you got kind of a byproduct. And this is called bilirubin. It gets converted into something called bilirubin. Bilirubin is another pigmentish. So if you ever have like a bruise, you always get those strange bruises that you find in the back of your leg, or around your knee, or on your elbow. You wonder what the hell happened there. Sometimes a bruise, which might be kind of blue-black for a period of time because of all the ruptured red blood cells, but a few days later, it might turn more of a yellow kind of mottled color to it. That would be the bilirubin that's kind of built up superficially in that integument in the skin. So it's got more of a kind of a yellowish look to it. So pigments, you with me? Ruby red blood, dark maroon blood, oxidated, deoxygenated. Here, even the leftovers of the heme group, when you rip the atom of iron off of it, has kind of a yellowy kind of weird look. So just a minor thing. We'll transport it down the liver. It gets converted into some substance called bile that helps us break down fats. This is projecting, so we'll come back to this digestive system. The bile is going to go into the digestive system, the small intestine, help break down some of the fats so we can absorb some lipids in the food that we eat. The bile moves on down through the small intestine, the large intestine. Bacteria say, bile, you're delicious, and they chomp it down metabolically, and they make something called stercobilin. And stercobilin is yet another pigment. It imparts the brownish coloration to the feces, just to be wonderfully graphic. So your red blood cells, you with me, ultimately give rise to the fecal coloration. How wonderful of a world is that? Do you need to worry about stercobilin for your first test? I don't think so, but you will hear this again when we get to the digestive system, so we'll harp on some of this again. Could I ask you on the first test, what does transferrin do? And I kind of expect you to say, oh, that's the one that helps take iron back down to the bone marrow, red blood cell recycling. Sure. You with me? But I'm not trying to get you with every little piece here. There's a little more of the story. We'll get to it in the digestive system. All right, so red blood cells. 45%-ish on average, male, females, right? 45% of the whole sample of blood, the hematocrit, the percentage of red blood cells, right? Around 45%, so that's a good component. 55% was plasma. We've talked about the water solution, everything that floats in it. Now let's come back to the Buffy code. Now this would be the formed elements or the cells, right? So the formed elements will include our platelets, which are called thrombocytes, and the white blood cells called leukocytes. So let's start with the leukocytes, and these would be the cells of our immune system. All of these are really more complete cells than like a red blood cell or a platelet, which I know we haven't really discussed yet. So these have a nucleus, they have organelles, they have a typical lifespan. Some only live a few days, a couple weeks. Some live for, a year-ish or more, but then they have the ability to divide to make copies of themselves that can exist in our tissues, indefinitely. A lineage, if you will, daughter cells, offspring, however you want to say it, that provide us maybe some memory for things that we recovered from, a couple, 3 years ago with COVID, maybe. You with me? So you now maybe have antibodies for whatever strain. I know it mutates all the damn time, but chicken pox, the flu virus from 1996, it may have, waylaid you for a week and a half. You still have those antibodies. That means you still have white blood cells that can produce those antibodies, you would think, that are gonna be in your system, so if you're ever exposed to it again, in theory, you got a little better chance at battling it before you get truly sick. Okay, so lifespan, all I'm trying to get to is kind of tricky. We'll flesh this out in another chapter. White blood cells may exit the bloodstream. There is a funky term, diapedesis. So diapedesis is something we'll talk about. All this we talk about later in detail. But if I say diapedesis, I mean a white blood cell can float in your blood, but it can also at some point say, I think there may be something causing an infection in the big toe. I'm traveling through a little capillary here near the big toe. It's time for me to leave the bloodstream and move out into the integumentary tissues to look for the site of infection, to kill the microbe that's causing you grief. So diapedesis is the process of them squeezing out through capillary pores. Leaving the blood is all I need you to know now. So diapedesis is a white blood cell says, screw it, I'm out of here, I'm leaving the blood, I'm going to patrol the body elsewhere. And that's what they do. If you talk about how they find pathogens, sites of infection, something causing you grief, so they can gobble them up or destroy whatever is making you sick. So think again, you stepped on a nail or something to toe, you have an infection. So how does the white blood cell find it? It has no eyeballs, you with me? How does it find that target? It's got little receptors on its membrane that are sensitive to inflammatory chemicals. So it basically can track, it's almost like a forked tongue on a snake, if you will, no tongue, but these little receptors, if you have more chemicals coming in from this side of the anatomy of the cell, right? That means the site of infection is isolated in this direction. If you have less of these chemicals interacting with the white blood cell on this side, they start to track. And this would be chemotaxis, tracking chemicals to find infection. Positive chemotaxis is more that way, I'm headed that way. So positive chemotaxis is not diapedesis, it's just saying how do they then, once they go through diapedesis, they're outside of the blood and the interstitial fluids, how do they find that site of infection? They're tracking chemicals through this positive chemotaxis. So all these we'll explain later. I don't need to go on and on about them. They're not the most important thing in this lecture to me, but sometimes I look at a little blurb on your screen and realize that I need to take a moment to flesh that out for you. So leukocytes come in two basic flavors. You've got granulocytes and agranulocytes. And you can see the little image there, agranular or granular. Granulocytes, agranulocytes to lack. So a granulocyte would be three types of cells, neutrophils, eosinophils, and basophils. We'll go through these in just a second. These are cells that when we take a blood smear, like if we get it from Carolina Biological Supply, they'll take samples of human blood, they'll fix it onto a slide, and they'll hit it with different stains. They'll hit it with kind of red acidic dyes or stains. and, go through the coloring process and rinse that off, and then they'll hit the slide, the blood smear, with kind of more of a purple basic. So red acidic pH, kind of a little more, low in pH, a little more acidity to it, usually reddish in coloration. Kind of the purpley blue, indigo, whatever you want to call them, are a little more alkaline or basic. The point is we do this in the laboratory, or they do it for us, and then they'll affix the slide, put the cover slip on it, it dries, and then when you and I look at it, we're gonna see white blood cells. Many of them are gonna have spots, grainy spots in the cytoplasm. So you usually can still see the nucleus, but it's in the cytoplasm, where you may see granules. And the granules are just little vesicles, little sacs, membranous-bound sacs inside. But if you have them, you call it a granulocyte. Some cells, called agranulocytes, don't have the spottiness to the cytoplasm. So they don't seem to show the granules. They don't have the granules. You with me? So this is all kind of about the way that we stain and fix the slide so you and I can look at it under a microscope. So all granulocytes, I say, can be phagocytic, phagocytic to engulf, kind of a big eater, if you will. It's endocytosis, where you kind of just ooze over, like, let's say, a bacterial cell, and then you just kind of pocket it in, kind of let your membrane surround it, and kind of bubble it internally. So now you have a little membranous kind of sac covering what you just ingested. But because it's a big thing, we often call it phagocytosis. So phagocytosis, think of one of these white blood cells ingesting some kind of a bacterial cell. Okay, so they're all capable of doing phagocytosis, but they all do things their own way. The first group's a little hard to see, the granules. So granulocytes would include neutrophils, basophils, eosinophils. Let me just show you real quickly two others so you can see them. So this is an eosinophil, and I can see kind of a bilobe telephone-shaped nucleus. Oh, you don't know what a real telephone looks like. Hello, hello, I'll call you. Do you still do that? Or do you say, I'll call you? Because you walk around like this and talk. I've seen you. Hello, you'll be talking, and I'll say, oh no, the cock's over here. And then I'll realize you're talking to somebody. Got one of them androids in your ears. You with me? That gets embarrassing. I said, well, I like salt and vinegar potato chips, but I don't know what you're asking. Then I realize they're talking to somebody. And then I just act like, I'm talking to somebody too. But at any rate, back in the day, don't you remember telephones? Do you remember the backlight telephones? I collect them and I try and rebuild them. I haven't done it in years. I've got a bunch though. The other ones go, bang, in the hole. It's a dial.
We have one.
Do you? You rock. Get your ass. Go on out. Okay. Air hugs, right? Yeah. But at any rate, I guess I say telephone shape, then I realize you don't know what the hell I'm talking about. So Google telephone, and then ignore the first 74 images. Okay? But at any rate, kind of a bilobe nucleus here. But look at this. My point to this is not to talk about any eosinophil, but I'm just saying, look at these red, coarse, grainy spots. Those are granules. You with me? So granulocytes, some cells are really easy to see. This is called a basophil. Look at that purple, grainy mess. Granules. You with me? So usually granulocytes, a couple of the classes are really easy to see. But we start off with the neutrophils, and it's not so easy to see. So just to show you, we're not done with neutrophils, but here's what we're looking at here. So all these are called neutrophils. And you have to look close, but I see faint little specks all out here in the cytoplasm. So this purple thing here, 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 and here, right? That's the nucleus. So granulocytes have granules which are, either faint or coarse, but they're little dots, or bigger dots, if you will, in the cytoplasm. So in neutrophils, unfortunately, the graininess is kind of faint. It may be the hardest to see, but it is there. You can't probably see it with resolution from where you are, but if you look at good screen images, and some of the ones I loaded up here I think are pretty good for you, you can see it. So neutrophils happen to be the most common white blood cell you and I will find in the bloodstream. So probably 60, 65%, upwards of 70%, 70 out of 100 white blood cells, if you're looking at a smear of blood, would probably be neutrophils. You with me? So they are by far the most common white blood cell. They're really key phagocytic cells. When we get to the immune system chapter down the road this semester, we'll talk about how these get released in huge quantities from the bloodstream if you're fighting off some kind of infection. And they will go and Pac-Man gobble up all kinds of microorganisms and viruses that are making you sick. So they're really important in terms of keeping us healthy. So 60-75% of all white blood cells, again, leukocytes, white blood cells, their diameter, look, I don't need you to memorize this, but you can just use references. So we said 7.5 microns is about the diameter for a typical red blood cell. Here we see that these are maybe not quite twice the size, but they're almost twice the size of a typical red blood cell, right? 10 to 12 microns. So there's an erythrocyte here. You see the biconcave, kind of the lighter color in the middle? That's where it's kind of pinched in the middle, no nucleus. So these are all my erythrocytes. This is all my plasma out here, and that's a neutrophil, that's a neutrophil, neutrophil, neutrophil. So do you need to know 10 to 12 microns? No. But do you need to know it's most abundant in the white blood cell, or white blood cell in the bloodstream? Yes. You with me on this? Okay, so you don't need to memorize microns. Go ahead.
Do the different shapes in the pictures that you have, do they matter, or is it just?
It's, all I'm trying to show you is multiple images because they all look a little different. And they can be a little confusing. So this is more for lab purposes, like in lab, whether you're with me in the 7-week that starts in October, whether you're with me in lab now, whether you're with Mans in lab now, or whatever. Some of you aren't in lab, you've already taken it, will take it. The point is that a lot of times you're looking at images and trying to name white blood cell, name a white blood cell. And you gotta look at a bunch of these because they all look a little different because their nucleus is, oh, it's down here, is, see this PMN? Polymorphonuclear. So their nucleus is highly variable. So it's often sausage link appearing, you with me? Like a little archipelago. It's like you took a long water balloon, right? And if you pinch it here and it drapes and bulges, and you pinch it here, you know, you can have kind of restrictions, but